Okay. Well, let's go in and get rolling for today. So, in terms of announcements, again, Quiz 6 is due Wednesday. I think that, depending on where we get today, I think we'll be mostly through the material for that quiz by the end of the day. So, keep that in mind. You have till Wednesday. Again, you get two opportunities to do that. Another group work assignment is coming up. It'll be either going to be this Friday or Monday. So it just really depends on how quickly we can get through Lecture 5 material, the material that we're currently working on. So it'll be essentially a start of Lecture 6, and Lecture 6 is glycogen metabolism. Okay, so and that's what the group work will be over, is breakdown and synthesis of glycogen. Okay? All right, so our riddle. What do you call an acid with attitude? A amino acid. Oh my goodness, okay. So great, right? So great. Okay, now, so actually here's what I want to go ahead and do today. I'm going to go ahead and start you off with a review question. We jumped into on Friday some of the particular details surrounding the regulation of glycolysis and gluconeogenesis, specifically in the liver. So I wanted to go ahead and ask you this question just to get things rolling for today, to get you back into your notes, looking at those things, remembering what we talked about, because some of this is we're going to pull forward into our discussion even today. So in review, which of the following is true? Phosphorylation of PFK2 FBPase2 by glucagon activates the kinase domain that makes F2,6BP. Phosphorylation of that actually activates the phosphatase domain that makes F2,6BP, or phosphorylation by glucagon activates the kinase domain that breaks it down, or activates the phosphatase domain that breaks it down. Okay, so what do you think? Go back and look at your notes. I think I'm going to give you 2 minutes for this one. Get engaged in this, and then we'll do a little review and head into new material. On your marks, get set, go. Okay, 2 minutes. What do you think? What do you think? About 20 seconds left, so if you haven't submitted an answer, do so. Okay, so let's go ahead and look at this here, see what kind of results we have here. So, okay, 62% say that phosphorylation of PFK2/FBPase2 by glucagon activates the phosphatase domain that breaks down fructose 2,6-bisphosphate. That that's correct. Okay, so that's good. You went back, you looked at your notes, or you simply remembered it from something else. That's good. So let me go ahead and come here. This is kind of the summary slide of that. It says by glucagon. It's really glucagon signaling. Glucagon itself doesn't do any phosphorylation. It leads to the activity of pKa by the production of cyclic AMP through adenylyl cyclase that is activated by the G protein that's activated by glucagon signaling. All right, so glucagon leads to that. All right. Now, that then is going to, pKa is going to phosphorylate a number of targets in the cell. Now, before we even get specific about the regulation of liver, and even specifically about that question, I want to just remind you that there is two things that are going to remain constant in our discussion on all these different tissues. First of all, fructose 2,6-bisphosphate is going to be an activator of glycolysis always. No matter what tissue you look at, it activates allosterically PFK1. It also inhibits allosterically FBPase1. Okay? So that's going to be true liver, heart, and skeletal muscle. Okay? The other thing that is also true in liver, heart, skeletal muscle is that this enzyme, the PFK2/FBPase2, that is going to have a kinase domain that will make fructose 2,6-bisphosphate and a phosphatase domain which will break it down. Okay, that's true in all tissues. Right now, specifically for the liver, glucagon signaling leads to the activity of pKa, which does indeed phosphorylate this. It phosphorylates, coming back here, okay, it phosphorylates this kinase domain. It's actually at serine 32, okay, the N-terminal domain of this. The number and identity of the amino acid is not important, but that is where it does this. And it then inhibits the kinase domain, activating the phosphatase domain, and this is what you then get. You get then the breakdown of fructose 2,6-bisphosphate, okay, by the phosphatase domain. That will then lead to activation of gluconeogenesis in the liver. Now, at the same time, something that I didn't mention is that glucagon signaling, and we're not going to go all the way down the rabbit hole on this one because it really gets into genetic expression, but glucagon signaling will also lead to increased expression of PepCK and glucose 6-phosphatase in the liver. Okay, so especially sustained glucagon signaling. So if glucagon remains in the bloodstream for a significant period of time, there will be an increased expression of PepCK, increased expression of glucose 6-phosphatase, and that means that the liver will slowly ramp up its capacity to make glucose through gluconeogenesis. Okay. Now, on the other half of this, insulin, this is what we ended with, and this was not, the question didn't have anything to do with insulin, but on the other half of this, insulin leads to the activation of a phosphatase, phosphoprotein phosphatase, that will remove that phosphate, activating the PFK2 domain, the kinase domain, and that will then produce fructose 2,6-bisphosphate, and that will lead to activation of glycolysis, and the liver will start to absorb glucose. Okay. Insulin, interestingly, okay, will inhibit, okay, the expression, genetic expression of PepCK and glucose 6-phosphatase. Okay, so those levels will start to drop when insulin, specifically sustained insulin signaling, is present. Okay. Now, these arrows here, this one, okay, and this guy right here, in regards to the activity of PepCK and glucose 6-phosphatase, I'm not going to describe those any further. Okay, we won't get into all the transcription factors that need to be involved in that. Okay, right. But there are pathways that have been worked out in that regard. Okay. So that's a review, for the most part, of what we had discussed on Friday. Okay. Are there any questions on this remaining from Friday and possibly even today based off of what I said? Okay, remember, it's always nice to take a step backward and think to yourself, okay, what is insulin saying to the liver, and what is glucagon saying to the liver? Okay, insulin says lots of blood sugar, the liver should absorb it. Glucagon says not enough blood sugar, the liver should excrete it. Okay, and then work your way forward from there. Right. Now, last thing to say here in regards to regulation of these in the liver, specifically in regards to what glucagon signaling can do. Specifically, and I'm going to go ahead and cut this slide right down the middle and highlight this over here. Glucagon signaling, again through the activity of PKA, will lead to the phosphorylation of the last step in glycolysis, pyruvate kinase. Okay. Now, there are different isoforms of pyruvate kinase. This book even mentions L and M. Okay, liver and muscle. Isoforms of this. You'll notice over here on this side of the slide, there's only liver highlighted here. Okay. The liver isoform of pyruvate kinase is sensitive to and is actually a substrate of PKA. Okay. And PKA can phosphorylate that, and that will activate it, so this would be a minus here. So again, signals from glucagon slowing glycolysis down. Okay. Right. Now, I think that it is important to say something in regards to my language there. Slowing it down is the way to think about this. Right? Very rarely do we see metabolic pathways truly switched off. Okay? So instead of thinking of them as like a switch, where you turn it off and on, it'd be better to think of it as like a dimmer switch, lower or higher. Okay, that's the way metabolic regulation works, is that rates go down, rates go up. Rarely do they just turn off. Okay, but in general, again, glucagon, okay, that's our signal here, is going to lead to a slowdown of glycolysis. Everything on the right we won't necessarily address right now. Okay, we might get to it later on, but I'm not going to address it on the right. Okay, and that's it in terms of what I want to talk about with the liver. Okay, so what I want to do now is step into skeletal muscle, and we'll go ahead and start this off. Not skeletal muscle, I'm sorry, cardiac muscle. Start this off with a question. So let's come here, go on to our next question. Thinking big now, stepping back, thinking big. What does epinephrine prepare the body to do? Okay, we're about to head into a discussion on cardiac muscle, but just generally speaking, epinephrine is a signal to the body to do what? Okay, generally speaking. I'm going to give you 2 minutes. Discuss that with your neighbor. Oh no, 1:45. Discuss that with your neighbor and submit an answer. What do you think? Epinephrine, what does it do? Okay, marks, get set, go. Okay, you got about 20 seconds left, so text me an answer if you haven't done so already. Got okay, about 5 seconds left. All right, let's check this out. Less responsive, but that's okay. All right. Epinephrine prepares the body for a fight or flight response. Is exactly right. Okay, signals a flight or fight response. Very good. Increased cardiac output triggers sympathetic nervous system. Very good. Okay, epinephrine activates the Hulk within you. I was hoping someone would put that answer down. That was the one I was really looking for. You're right. It makes you big and green. and angry, right? No. Okay, but yeah, there's, I mean, a number of good responses. Increased heart rate to survive by running or away or by fighting. Exactly right. Something to that effect, okay? That would be just generally enough what I was looking for. Epinephrine, also called adrenaline, is going to prepare you to do something that maybe you wouldn't normally do. If you saw a bear that started to run at you, or let's go a little bit more. Did you hear about the guy that got attacked by the mountain lion in Horsetooth Park and suffocated the mountain lion? Epinephrine was his friend. Let me tell you something, okay? Fight or flight, okay? And he decided the best way to do this is to kill the mountain lion, okay? Which is a remarkable story that made national news. My dad sent it to me. He was like, wow. And he lives in California. Like, was this you, Aaron? And I was like, no, but kind of. I mean, you know, I was almost there. So that's what I would have done. Okay, anyway. So fight or flight, exactly right, current news. Now, when you think about this, so let's go a little bit more on the serious side of things. Let's come back here. Okay, I want to talk first of all about cardiac muscle, and we'll also talk about skeletal muscle too in terms of regulating glycolysis and well, really glycolysis is what we'll spend more time thinking about. So cardiac muscle and skeletal muscle are very similar in some ways, and so I'm going to highlight some of those similarities in terms of the regulatory capacity of glycolysis, but they're also different. Okay, and we'll start with some of the differences here, starting with cardiac muscle or the heart. All right, and epinephrine is one of the signals that can lead to the regulation of glycolysis, in particular an increase in glycolysis. Okay, like we're saying, fight or flight is going to lead to really increased heart rate. You have to actually go through vasodilation, these types of things. You're preparing your body to do something strenuous. Okay, and so certainly the heart rate is going to go up. You're going to contract. There's going to be more contraction, and therefore, to support that increased contraction of the heart, you're going to have to increase ATP producing capacity in the heart. Okay, so glycolysis then and the rate of glycolytic flux should go up. Right, what you are looking at here is again the kinase phosphatase bifunctional enzyme, PFK2. Right, so we have here PFK2, and we have here FBPase2. Right, all in the same enzyme. Now, here's what's really fun, okay, about this whole thing. This is a different isoform of this enzyme. Okay, you'll notice here it says 530 amino acids. If I go back several slides when we were talking about the liver, it was 470 amino acids. And I started off today saying, oh, there's this phosphorylation site, serine 32, at the front end of this molecule. Well, come to cardiac muscle, and this is a 530 amino acid protein, and there are two phosphorylation sites, and they're both at the C-terminus. There's actually one in the kinase domain, though we don't really quite understand exactly what it does, so don't worry about that. But the phosphatase domain we've got pretty nailed down. And I think that these are serine, threonine, 466 and 475 or something like that. Okay, but C-terminal domain is what you need to remember. Okay. Now here's what's really fun. Those inhibit the phosphatase domain. So epinephrine is a very similar signal to glucagon in that it leads to the activation of adenylyl cyclase through a G protein that leads to the activation or the production of cyclic AMP that then leads to the activation of pKa. That then in the cardiac tissue though will phosphorylate this isoform, but it's going to phosphorylate the C-terminal domain. And it's going to inhibit the phosphatase domain. So that means this, okay, happens. Again, we got sunbeams. Sunbeams come out of this, okay, and we can come here and have this kind of a slide. Don't worry about AMP kinase quite yet. We'll get to that here in just a second. Okay? But what's happening here is this. This is, again, pKa. pKa leads to the phosphorylation. We could really put another phosphate group here because there's two phosphorylation sites. Okay? And that is going to lead to the activity of PFK2, which is going to then do this. Let's do it over here. Fructose 6-phosphate, fructose 2,6-bisphosphate, and that's going to increase glycolysis, plus glycolysis. Really, it's PFK1. Okay? And epinephrine, very capable of doing that. And that will then support increased muscle contraction, which is important if a mountain lion does indeed attack you in horsetooth. Okay. All right? So, now notice what I'm saying here. This is the exact opposite of what happens in the liver. Okay? And yet the signaling is, virtually identical. Okay? The biggest difference is that it's not glucagon, it's epinephrine that's causing the effect. Okay? But everything else that I said, right, all the names are really similar, okay? And even the pKa that phosphorylates this, but the result is the opposite. Now, cardiac tissue, cardiac muscle doesn't have a receptor for glucagon, okay? And that's a good thing, because every time your blood got low in terms of glucose, then your heart rate would increase based off of this, which means that when you woke up from a normal overnight fast, your heart rate would be high, and that would not be great. Some of you are like, actually, it might wake me up. So it's kind of what caffeine does, doesn't it? Yeah, but you don't want this on a daily basis, so again, not good. Okay. All right, but so keep that in mind, okay? And you're going to have to keep this straight. This is biochemistry for you. Okay. Now there are some other things that I want to go ahead and mention here, and this is going to bring up a really important topic. So I'm going to draw this. We're going to go in the exact same direction. There's actually another enzyme, and I kind of crossed it off over here, and I'm going to go ahead and put it over here. AMPK is what it commonly goes by. Okay, and this could be also considered AMP activated kinase. That would be another name for it. So AMP activated kinase. AMP activated kinase, or AMPK, is a really important regulator of metabolism, in particular of catabolism. Okay? As the name suggests, is activated by the molecule AMP, which is a breakdown product of ATP. Okay? And so AMP kinase will activate, it's going to do the same thing. It targets this PFK2 FBPase2. It targets the C-terminal domain, and it will do this exact same thing. Okay? Strangely, it only phosphorylates one of the sites. Okay? Epinephrine pKa will lead to phosphorylation of both sites. AMP kinase only one site, though the overall effect is the same. Okay. Now understand then, if we're looking at this, okay, so AMP, that's our molecule. This is going to activate this. Right? This goes up. AMP goes up when there's sustained contraction, okay, or increased levels of contraction. So let me do this for us here, okay. Where to write this? Let's do this. I'm going to box this off. If you've studied contraction of muscles, you know that, and I'm not going to do this, but ATP is used to contract muscle, okay. ATP, though, will be broken down in contraction of muscle to ADP and inorganic phosphate. Now here's what's fascinating about this, is that there's another enzyme that can take another ADP, okay, and convert two ADPs to an ATP and an AMP. Okay, so this is true in skeletal muscle. This would be true in cardiac tissue. All right, anytime ATP is hydrolyzed to ADP, there's another enzyme called adenylate kinase that will take two ADPs and make an ATP and an AMP molecule out of it. So if you have increased contraction of these muscles, then you'll have increased levels of ADP. This is a reversible step. The enzyme adenylate kinase holds this reaction in at equilibrium, and if these are going up, then this is going to shift in this direction, and you'll have an increase in AMP. That will lead to more of this. That will activate AMP kinase, which will activate glycolysis, which will activate the capacity to then make more ATP. Okay. Now the name of that enzyme, let me go ahead and write that down here. Of course, I'm running out of room, but I'll draw it over here. This is adenylate kinase. And it's not only found in cardiac muscle or skeletal muscle, it's found in all tissue types. Okay. In every single one of your tissue types, you have this molecule, adenylate kinase. Okay. That can do this. And it's reversible in all tissue types. Okay. So it can go in the opposite direction too. All right, so that's some of what we'll say about cardiac muscle, okay? We'll talk about some of the similarities it has with skeletal muscle, but this would be the biggest thing here. Obviously, it's very different from the liver in many respects. Okay. Questions on this? Yes?
If the adenylate kinase is able to produce ATP from ADP, and you break down ATP in cellular processes, and then if you build it back up through glycolysis and the rest of that pathway, why isn't this considered a pathway to produce ATP?
Okay, so the question is, in regards to if you have adenylate kinase that can take these ADPs and make ATP from them, why is this not considered like one of these pathways that can make ATP? In some regards, it could be considered a step. I mean, it's not a pathway, but a single step to make ATP. It's more of a break even, though, than anything else, because you have to have the ADP around. So you can't take, for instance, like glycolysis, which would be true catabolic pathway, where you start with a carbon-rich molecule, start moving electrons around and off of that to make ATP. This requires some level of ADP in the cell. It's not going to get any energy from anything else but ADP. Does that make sense? Okay, so it's not harvesting energy from something. So it's more of a break even. Okay, because what will have to happen, just so you know, that AMP will have to be converted back to ADP using the ATP molecule. Okay, because most processes that make ATP require ADP to do that. Okay, so other questions? Yeah, is that a question? Oh no, okay, you're just itching. Okay. Now let's do this. Okay, real quickly. AMP kinase, we need to talk a little bit more about that. This is the first time we've been introduced to that, but let's go here to give you an idea. And this is big picture now. Okay, I'll kind of cut this one in half too. Okay, and we'll start looking at the top half of this, and then we'll kind of move to the bottom half for a little bit, but not that much to look at down there. Okay, so AMP kinase sits in the middle of this here. All right, and I'm going to go ahead and highlight this exercise. So, obviously you're working out, some sort of strenuous activity is going to lead to increased levels of AMP in the skeletal muscle, increased levels of AMP in cardiac tissues and other tissues as well. All right, that can activate this molecule AMP kinase. And what I'm going to show you is this. You look at these two things, okay? We just talked about how it can lead to the activity of PFK2. Okay, that PFK2, FBPase2 is going to phosphorylate the phosphatase domain of that, and that will lead to increased cardiac glycolysis. Okay. It will also do this. So we talked about how GLUT4, I'm going to highlight just GLUT4 here. Okay. Well, we talked about how GLUT4 in skeletal muscle and adipose tissue can be mobilized to get into the membrane of those tissues when insulin is present. And it's in those vesicles. Right? AMP kinase will take a different route to do that, but it will do the same thing. Okay? It will activate the mobilization of those vesicles to put GLUT4 into the tissues of, in particular, muscle and adipose. And those tissues will start to absorb glucose from the bloodstream. Okay? And of course they need to, because if AMP kinase is activated, then that means that energy supply is low. Now let's go here, and we have yet to really build the groundwork for this, but we will do this. And I'm just going to highlight some things here, and I'm just going to highlight the word synthesis in all this. Okay? AMP kinase is going to inhibit most anabolic processes in the cell. Okay? So essentially, AMP kinase says, whoa, there's a very low energy supply. We need to get energy into the cell through glucose, glycolysis needs to increase, and everything else that's using ATP needs to stop. Okay. Now down here, this would be specific tissue distribution of that. Maybe we'll build the story as we move along. In fact, I'll bring the slide up a couple of times throughout the semester. Okay. But this is our first introduction to AMP kinase, very important regulator of lots of different pathways in the cell. I will highlight this one thing here. Okay. It will also increase activity of fatty acid oxidation, which is also a major means of producing energy in cells. Okay. These enzymes here, when you look at these, right, we haven't yet really talked about any of these yet. Okay, and certainly we'll talk about some of them, not all of them. We won't really get to protein synthesis at all, so we won't talk about that at all, but we'll talk about some of these others. Okay. Questions on that? All right, AMP kinase, very important, kind of a central regulator of catabolism, if you will. All right, now, let's do this. Let's step from cardiac muscle, having highlighted essentially epinephrine and, of course, the AMP kinase, and let's move to skeletal muscle, and let's talk about, first of all, the differences it has with cardiac muscle, and then we'll talk about the similarities that it has with cardiac muscle. Okay. All right, so here would be, here's the reaction catalyzed by PFK1. Okay, PFK1 catalyzes third step in glycolysis. And so this enzyme here that's not listed is PFK1. And of course, you know, it catalyzes ATP-dependent phosphorylation of fructose 6-phosphate, making fructose 1,6-bisphosphate. We'll talk about the top half of this in a little bit. I want to focus on the bottom half for right now, though, because I want to complete our little discussion on this guy right here, fructose 2,6-bisphosphate. We have been discussing how fructose 2,6-bisphosphate can either be made or broken down by the kinase domain or the phosphatase domain of PFK2, FBPase2. Skeletal muscle has that same enzyme in it. I guess I shouldn't say that same enzyme in it. I don't have a devoted slide to this, but I'll go ahead and draw this up. Something very similar anyway. It has this enzyme that has a PFK2 and an FBPase2 domain. However, unlike the liver and unlike cardiac muscle, this one cannot be phosphorylated. Okay? So there's no phosphorylation of this enzyme in skeletal muscle. Right? In fact, the isoform of this is the exact same isoform that you find in the liver, but missing the N-terminal domain, which is where the phosphorylation site is for the liver isoform. Okay. Now, this again, right, can take fructose 6-phosphate and make fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate will activate PFK1. Nothing new necessarily there. This will also then take fructose 2,6-bisphosphate and break it down to fructose 6-phosphate. The question is, well, what regulates that? If it's not phosphorylation, there has to be something. Well, it turns out that this molecule here, fructose 6-phosphate, will allosterically activate PFK2. Okay, so allosteric activation of PFK2, the kinase domain. Okay. So if we consider this, you have skeletal muscle that's absorbing glucose. Maybe it's using ATP. If it's absorbing glucose, hexokinase will catalyze the trapping reaction that will increase glucose 6-phosphate. Traps it. Phosphohexosaisomerase, the second enzyme in glycolysis, holds glucose 6-phosphate and fructose 6-phosphate in equilibrium. So if glucose 6-phosphate concentrations are going up, fructose 6-phosphate concentrations are going up. That will feed forward and activate PFK2, which will make fructose 2,6-bisphosphate from fructose 6-phosphate, activating PFK1, activating glycolysis, so that ATP can be generated by the glycolytic pathway. Okay. On the opposite end of this, some of you are looking at this saying, well, okay, wait a second. Fructose 2,6-bisphosphate, though, gets broken down by FBPase2 to fructose 6-phosphate, which activates this, right? And you start to think, that can't work. How does that work? Wouldn't that be the problem? Well, here's the deal. If this is being broken down, you're not allosterically activating this any longer, right? Being broken down to fructose 6-phosphate, signals in the cell are going in the reverse direction, right? This will be actually being converted back to glucose 6-phosphate, and in skeletal muscle that will be converted to glycogen. Right, so it can get fairly hairy fairly quickly, unfortunately. Okay. So no phosphorylation here, only allosteric activation. Questions on that? No? Okay. You just need me to give one more acronym that has PFK in it, and that will be great. Or F6P or something like that. Okay. All right. Now, that is certainly one of the means of regulating skeletal muscle. Different than cardiac muscle. Cardiac muscle is sensitive to phosphorylation of that enzyme, and of course different than liver, which is sensitive also to phosphorylation by different signals. Okay. Skeletal muscle doesn't really have those types of signals in terms of phosphorylation. And that kind of closes the book in terms of PFK2, FBPase2. 3 different tissues, right? 2 different phosphorylation sites and 2 different tissues, and 1 with no phosphorylation site and only allosteric activation. Now, what I want to do is move to the top of this and talk about this. Okay, because in skeletal muscle, this is actually the most important aspect of regulation of PFK1 and glycolytic flux in many ways. And that is the role that ATP and its breakdown products ADP and AMP play. Now again, ADP, 2 ADPs equal an ATP and an AMP. Okay, so we've got that hopefully in our minds. So we're going to look at that, and everything that I'm going to say about this in terms of skeletal muscle would also hold true in cardiac tissue. Okay, the muscle tissues, the PFK1 is very similar in both of those, and they can both have this type of activity related to ATP, AMP, and ADP. Okay, and so let's look at this. We'll start here looking at ATP effects. ATP actually, okay, is an inhibitor. Yeah, actually, we go to this slide here. There we go. Is an inhibitor of PFK1 activity. Okay, and here's a graph that's in your book showing this relationship. Okay, so we have PFK1 activity here versus its substrate, fructose 6-phosphate. Now remember, this enzyme catalyzes this reaction. I'll go ahead and write it up here. Fructose 6-phosphate plus ATP. Okay, goes to fructose 1,6-bisphosphate plus ADP. Okay, and we see here 2 different curves with 2 different levels of ATP, high and low. And you'll notice that in the presence of high ATP here, the activity of PFK2 goes down. Not PFK2, PFK1, sorry. You say, oh, see? I got, okay, fine, I'll give myself a red check minus. Okay, there we go. I misspoke for just a moment. Right. Now, when low ATP activity goes up, now understand what we're saying here. Let's do this. Let's assume fructose 6-phosphate levels are going to remain at some steady state. There's always a range that these concentrations go in. Okay, so we'll just pick one of those out of the range. But let's just say that this happens to be the steady state concentration of fructose 6-phosphate. Okay, So steady state concentration of fructose 6-phosphate. Notice that in level, in high ATP, notice what we're saying here. We have somewhere around, I don't know, 25% of the Vmax. Okay, but if we were to come all the way up here, when ATP levels drop, then we're sitting somewhere around like 90% of the Vmax. Okay. This huge discrepancy between the two. Right. High ATP, again, would be the signals from the skeletal muscle or in the skeletal muscle saying there's no need to break down glucose to make more ATP. Okay. So it would decrease or diminish activity of course, PFK1. And honestly, there would be other enzymes that would be sensitive to this in glycolysis, but flux through glycolysis will go down. ATP levels change and you have this big jump. Okay. And it can be a really fast jump. Okay. They're very sensitive and is very quick to respond in this way. Now, let me ask you a question. Okay, last question of our day today. Considering what I just told you, ATP as the substrate of PFK1 should lead to an increased catalytic rate. How then does ATP lead to a decrease in catalytic rate, right? So everything that you've learned about enzymes and everything that you learned about rates of reactions, if you have a reactant going up in concentration, that should lead to an increased rate, right? And yet what I just showed you is that ATP leads to a decrease in rate. How can that be possible? So I'm going to give you 2 minutes. Discuss that with your neighbor. Text me an answer. Okay, I can answer this in 2 words. You don't have to answer it in 2 words, but I can challenge you. Okay. All right. So, what do you think? Talk to your neighbor about that. How is this the case? About 30 seconds left, so submit an answer. It'll take a little bit of time to text these or type them, so submit an answer. All right, 10 seconds. Okay. Let's check it out. See what people put here. Okay, so, allosteria. Oh, 1 word. Very good. Allosteria. Okay. All right. Just does. Or just because, right? Something like that. This person must be having a bad day right down here, right? Okay. All right. Death. All right. By lowering the concentration and producing negative feedback, feedback inhibition. Negative feedback loop, okay. Blocks binding site for allosteric activity, okay. These are the 2 words that I would have used. Allosteric inhibition. Okay, so the word, that's what I was showing you on this graph, okay. It certainly, like negative feedback loop, that's exactly what this is. But negative feedback loop by allosteric inhibition. Okay, this describes kind of like a large process. How does that happen? Usually through allosteria, okay. Not sure that's an okay answer. Is even 1 word, right? 1 syllable even. All right, now, Brevities. Okay. All right. So coming back here, this is what I'm showing you here. Okay. And this was to just remind you. Now we're applying what we learned all the way go back to lecture 2 when we talked about allostery. And I gave a graph real similar to this. Okay. Just had one where there was no allosteric modulator, and then we had allosteric modulators chalked through it. Okay. Going positive or negative. All right. That's exactly what this is. So ATP is not only the substrate of PFK1, it's also an allosteric inhibitor of the enzyme. Right, so if we come here, let's look at this. This will remind you a little bit of what you would talk about in like BC 401. We have to have some like structural analysis of this. What you see here, this is PFK1. PFK1 is a homotetramer. Four subunits, identical. Okay, you see these 4 subunits here. They're colored in different shades of blue and gray, I guess, here. So subunit 1, 2, 3, and 4. Each one of them they have ADP bound in this crystal structure, okay? But each one of them would have an active site. Okay, you can't see the other 2 active sites. They're on the opposite side of the molecule, okay? But these would be part of the active site. And you can see also here fructose 1,6-bisphosphate is there, okay? So this is active site. Okay. Now, in addition to the active site, there's also this site, and it's sandwiched between the subunits. And so you can kind of see that they have this outline of the molecule here, but there would be this allosteric site, okay, or a regulatory site. And it's found on, it's a little bit better view of it. Okay, it's found kind of sandwiched in between them. There's actually 4 of them all together. Okay. And ATP can bind there. And when it's bound there, it will favor or cause this molecule to favor the T state. Right? T state meaning low activity. Okay? Anytime you have allostery, we have T and R states. Right? And if T state starts to become favored, that's because there might be some allosteric inhibitor or modulator involved in this, and that's exactly what's happening here. So allosteric inhibition is how we explain this. Now, if we're to do this, let me go one step further here. And let me go ahead and draw out something here. If we were to plot percent of Vmax, okay, so rates essentially on our y-axis, and this would be percent of Vmax of PFK1. And we are to plot it versus the concentration of ATP. So we're going to hold fructose 6-phosphate concentrations stable in this experiment. Okay, and we're to then take different percents of Vmax at different ATP concentrations. What you would see is that at really low ATP concentrations, okay, you'd see this increase in rate up to a certain point, and then it would reach this peak, and it wouldn't be quite 100% of the Vmax, but then it would start to taper off, okay, in terms of PFK1 activity. And so what we have here is this increasing rate. And then we have this decreasing rate at some ATP concentration. And this can all be explained by the relative affinities of the active site versus the regulatory site for ATP. Turns out that the regulatory site has a lower affinity for ATP than the active site. Okay, so there are certain ATP concentrations when it's low enough where ATP really is only occupying the active site. A very small amount of it is occupying the regulatory site. And so the majority of the molecule will then be in the R state. And as ATP concentrations go up, you'll see this increasing rate. However, as ATP concentrations get large enough, pretty soon it will bind not only the active site, but it will also start to occupy the regulatory site. And of course, the more occupancy of the regulatory site of ATP, the more the molecule will be in the T state and the less the rate will be. Okay, so you'll see this decrease. Right? And this is, I mean, PFK1 is a great example of this, but this is it's common. I mean, it's not something that is abnormal to see where you have, let's say, an enzyme that has a substrate that can also act as an allosteric activator or inhibitor. Okay? And it's not, especially when the enzyme uses ATP, okay? Because ATP is a very important allosteric activator or inhibitor of enzymes. Okay? Let's go ahead and stop there. That was a load of information today. Okay, so we'll stop there, come back on Wednesday, do a little review, and then move forward. Okay? All right.